Cell cycle and cell division. Meiosis. Meiosis. By sexual reproduction, the production of offsprings includes the fusion of two gametes, each with a complete haploid set of chromosomes. From specialized diploid cells, gametes are formed. This kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This type of division is called meiosis. In the life cycle of sexually reproducing organisms, meiosis ensures the production of haploid phase, whereas fertilization restores the diploid phase. In plants and animals, we come across meiosis during gametogenesis, which leads to the formation of haploid gametes. The following are the key features of meiosis. In meiosis, only a single cycle of DNA replication takes place but involves two sequential cycles of nuclear and cell division called meiosis I and meiosis II. After the parental chromosomes have replicated to produce identical sister chromatids at the S phase, meiosis I is initiated. Pairing of homologous chromosomes and recombination between them takes place in meiosis. At the end of meiosis II, four haploid cells are formed. Under the following phases, meiotic events can be grouped. Meiosis I involves prophase I, metaphase I, anaphase I, and telophase I. Meiosis II involves prophase II, metaphase II, anaphase II, and telophase II. Meiosis I, prophase I. When compared to prophase of mitosis, prophase of the first meiotic division is typically longer and more complex. Based on chromosomal behavior, it has been further subdivided into the following five phases, that is, leptotene, zygotene, pachytene, diplotene, and dikinesis. Under the light microscope, during leptotene stage, the chromosomes becomes gradually visible. Throughout leptotene, the compaction of chromosomes continues. This is followed by zygotene, the second stage of prophase I. Chromosomes start pairing together during this stage, and this process of association is called synapsis. This kind of paired chromosomes are called homologous chromosomes. Chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex as indicated by electron micrographs. The complex, which is formed by a pair of synapsed homologous chromosomes, is called a bivalent or a tetrad. At the next stage, however, these are more clearly visible. Compared to the next stage, that is pachytene, the first two stages of prophase I are relatively short-lived. Bivalent chromosomes now clearly appears as tetrads during this stage. This stage is marked by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. The exchange of genetic material between two homologous chromosomes is called crossing over. Crossing over is also an enzyme-mediated process, and recombinase is the enzyme involved. On the two chromosomes, crossing over leads to recombination of genetic material. By the end of pachytene, recombination between homologous chromosomes is completed, leaving the chromosomes linked at the sites of crossing over. The dissolution of the synaptonemal complex at the beginning of diplotene is recognized by the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. These X-shaped structures are called chiasmata. Diplotin can last for months or years in oocytes of some vertebrates. Dikinesis is the final stage of meiotic prophase 1. This is marked by terminalization of chiasmata. The chromosomes are fully condensed during this phase and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. The nucleolus disappears by the end of dikinesis and the nuclear envelope also breaks down. Transition to metaphase is represented by dikinesis. Metaphase 1. On the equatorial plate align the bivalent chromosomes. From the opposite poles of the spindle, the microtubules attach to the pair of homologous chromosomes. Anaphase 1. The sister chromatids remain associated at their centromeres while the homologous chromosomes separate. Telophase 1. The nucleolus and nuclear membrane reappear, cytokinesis follows, and this is called a dyad of cells. In many cases, although the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic divisions is generally short-lived and is called interkinesis. Interkinesis is followed by a much simpler prophase than prophase 1 called prophase 2, meiosis 2, prophase 2. Usually, before the chromosomes have fully elongated, meiosis 2 is initiated immediately after cytokinesis. Meiosis 2 resembles a normal mitosis in contrast to meiosis 1. By the end of prophase 2, the nuclear membrane disappears and again the chromosomes become compact. Metaphase 2. At the equator, the chromosomes align at this stage and from opposite poles of the spindle, the microtubules gets attached to the kinetochores of the sister chromatids. Anaphase 2. Of each chromosome, it begins with the splitting of the centromere, allowing them to move towards opposite poles of the cell. Telophase 2. With telophase 2, meiosis ends where once again the two groups of chromosomes get enclosed by a nuclear envelope. Cytokinesis follows, which results in the formation of tetrad of cells, that is 4 haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which of each species conservation of specific chromosome number is achieved in sexually reproducing organism across generations, even though the process per se paradoxically results in reduction of chromosome number by half. In the population of organisms from one generation to the next, it also increases the genetic variability. For the process of evolution, variations are very important. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.